we get experimental science. We're curious, non-judgmental.
So, what is the new species of the week? Now that we see it, chat, the resounding fish. So, this is the title of the new species of the week. And chat, what's really cool about this is also the saddest part of this paper, chat. So, when I first read this, I just looked at the title and I was like, huh, what is this? This was like the species with the name, because it looks a little bit like Megalodon, right? Turns out it's not Megalodon like the shark. It is like a weird derivative name of Megalodon that gives this extinct species its name of Malagodon. So I was stirring the letters a smidge there. It is by no way called the Megalodon. I would prefer it to be a Megalodon, but it is not. It is the Malagodon is the name of this fish. And it was discovered in Madagascar, of all places, in 1994. Now then, Sam, are you ready for the sad part? In 1994, no one thought this was a new species. Everyone was just like, oh look, pretty fish. And they took this pretty fish to a museum. Let me show you the image of pretty fish. There is pretty fish. It's a very, very beautiful fish. They took it to a museum and they tried to propagate it. You know, that's what they wanted. They were like, oh, let's get more of this fish. And there was no real identification with it. The researcher was like, there is this fish. It was found on the island of Madagascar. Apparently swims near the top of the water where it's the water is very clear. And apparently there's a lot of reflection. And they have notes from that time where that demonstrate there's a bunch of these fish there, fishermen. It wasn't just one they found. There's a bunch of them swimming on around, and they see that the light they're right underneath the surface of the water. So at the top of the water, these multicolored fish are swimming around. And their researchers are like, oh, there's a bunch of reflections that are going here. Like, that's really cool. What they found, though, when you got deeper in these, like, murky waters, and actually, fishermen, I have an image of what it looked like of where they found the fish. This is where the fish were found. This is the kind of setting that we're looking at here. So you can see it's inland, it's a body of water, it's not super deep to begin with. And as you're looking at this body of water, right on near the surface is where you're going to be seeing these fish. Yeah, and the trees are, I mean, they are beautiful, right? Like, these are like the top tier kind of trees. These kinds of waters are where this fish was found. And guys, I'm going to put back up the fish just because I think the fish is really, really pretty. And this is also why the fish was noticed, right? Because it's so brightly colored. So again, this was in 1994, and they grabbed one of the fish. So Golganak, what I think is really cool with the fish is that, and the researchers, they were like, well, we don't know, like, how many they are. And so they were just like, well, let's only take a few. So they took about 10. They took about 10 of these back to the United States. They took them to the University of Michigan. They just kept it in an aquarium, and they tried to breed it. They fed them with, and no idea knowing what the diet is, okay? They fed them algae, zooplankton, and larvae. And they lived for 3 years, didn't reproduce once. Fishman didn't reproduce a single time. That was it. And so what ended up happening? Researchers preserved the fish. They put it into an ethanol specimen and made sure that it was kept preserved so they'll be able to, to look at them in the future if they wanted to. And so fast forward, y'all. Fast forward 2023. There's a scientist in the lab, and they see this vial with the fish. And a little bit what they see is like, this fish looks a little strange. Did they catch all, ladies, or as Golganak said, all of the brosifs? That is not what they noticed. What they noticed is, and you can kind of see it here, is you're seeing some leak through in the image of skeletal structure. You can kind of see vertebra a little bit. You can see the vertebra a little bit, right? This fish lab, this researcher in 2023 is like, something about this seems really off. And the thing that seemed off about this was the number of vertebra. So they actually did a stain. So actually if you actually look at the behind the scenes stuff and how they describe the study, they actually did these experiments very much out of order. And so the first experiment they did was on this side of things where they actually did the stain. So what they're doing is they took this preserved fish, they took a bunch of different images of said fish, like in its natural state. And if y'all want to, I can show you. It was after, I guess, what was it? 97, 07, almost 30 years, the colors have faded. So this is what these fish look like now. And actually, they looked back at these two samples that they had preserved in the alcohol, and they noted that there is male and female. They were able to sex them based on similarity of other fish species. And they're like, okay, these look near identical in terms of like how to sex them. Cool. We had males and females. That's not the weird part. But they did notice in the old images that were taken, there's something off with the vertebrae. They were adults as well, Sam. They were not juvenile states. That's more of an allegedly. But then they did this clearing process. So this process is a means by which they make essentially everything transparent in the fish. And then they stain the bones and other developed bits with different chemical dyes so that they can get a nice image underneath the microscope. And the reason this is important is because it really lets you firmly visualize what is happening. And so here, the thing that's happening is you have an additional vertebra. So for all the samples that they already had specimens of in the lab, they found this extra vertebra. So it is not a one-off. I know Smikes will tell us, and he's very accurate, like, oh, this only happened one time. And that's always a good call out. If something like this only happens once, is it really relevant? Is it biologically relevant? And the answer is probably not, right? You need to have this be represented multiple times to show that it's not just a one-off mutation that happens, but in fact is a biological phenomena of importance. And so what they find is every sample that they had preserved, they did this analysis on, had this vertebral segment. Okay. So that tells us, and by extra, I mean everything classified from that area does not have this extra vertebra, right? So this is a developmental difference in these creatures, which again, I'm like, that is amazing that there's this level of detail that we're able to get. Okay, so super, super cool. Where does it get a little bit, maybe a little bit on the sadder side of things? So they went back to Madagascar. They go back to Madagascar and like, all right, let's try to find this fish. We've already done morphological characterization of it. We have this old data demonstrating where they ended up sleeping, like in terms of like surviving in the water. Let's get some new samples and do some sequencing. They go back, there's no more fish. There is not a single brightly colored fish as was in the images taken. And on like these little swamps in Madagascar. And so where it really turns out is that these researchers had found back in 1994 a brand new species of fish, never been described, and only almost 30 years later someone was like, hey, this preserved specimen on the shelf looks a little different. Like, let's take a close look at it. And so I thought this was kind of interesting because they took existing data from the 90s and modern day reanalysis in terms of morphology on this and took a look at the fish, and they actually named it this Malagodon Honahona. The Honahona is this native language called Malagasy, and it means swamp. So I actually named it after the location that it lives in, which is a swampy-like area. When they did these other comparisons to existing fish that are still there, so Golganite, it's not that there's no fish there anymore. The deeper dwelling fish that are in the murkier waters that are darker, turns out those creatures are still there. Which is again like, shoot, why are those? Yeah, so exactly, fisherman. It is a new extinct species of fish. It's not even food anymore. So it's the standard way of doing this, I guess, which I like your approach much better, Cliff. They actually didn't do any sequencing in this. Because there was no sequencing in this study, Cliff, one could argue that you couldn't do the eDNA test because you didn't have a genome to compare against, right? So if you haven't sequenced this fish, you'll have no idea what eDNA you're kind of grabbing. And for those of y'all in the chat who are wondering what the heck is eDNA, is that like RNA? eDNA is environmental DNA, where you can just essentially sample from the air that just has like DNA floating from different creatures. And if you just analyze them at a different concentrations, you'll be able to find out who it belongs to. Because there is no sequence associated with this creature, they wouldn't be able to do that. So the most I think they could do, Cliff, is maybe if they did the eDNA test and there was a mystery species there, then maybe that mystery species could be the species that they are interested in. But, right, there could be other mystery species that aren't sequenced as a possibility. Now, what they ended up doing, Cliff, is the standard argument, I guess, is look when surveying an area and looking at percentages, so how much of a given area that you're surveying has this kind of fish. Sam wonders if there could be the wrong location. So the notes that they have from the 1994 expedition of where they collected them even has a map. So unless they were super, super off, they had data of exactly where to go. And there wasn't like a sense of where exactly where they shouldn't go. Like, so this is the map here. This is obviously a modern day figure, but they were looking at this red dot below. That is the new species location. There is this upper dot here of an existing fish that is still alive today. And they actually went and sampled both locations, Sam. If it was a suggestion there might be on a different island or like on a different like setting of the island, that's a possibility. They suggested that, again, the measurements of the density would suggest that there's none in those particular, because there's none there would be none elsewhere as well. So that I thought was a little interesting on that. Maybe, Sparky. Maybe there was one off to a different area, but they're suggesting that it's small enough, like the island, where because it's small enough, if they're not in these endemic areas, where these locations here that they are marking are these kind of like deep, like style of water where they were originally discovered, that was the suggestion of like maybe that's not what they're there. So Coffee Rocks is asking, like, couldn't they just get some DNA extracted from the ethanol preserved specimens? They could. There just wasn't done any in the paper, Coffee. So, like, to get Cliff's question of doing the eDNA sequencing, you'd have to go ahead and do the sequencing on those specimens that you had, and then move to the sequencing, like the environmental one. But they did neither of those. And I don't know, again, if that's a budget thing, or I will give you a quick verbal summary, and then we can actually look in the paper if you would like. One is, so looking at this coastal forest environment, that coastal environment they suggest has been washed away. So it's not just like a degradation where like sand leaving, but there's been like just ice, like the whole region's been isolated, right? So like upper areas, it's like deeper in the forested areas where some of these were located, are now dried out, and then once closer to the shore have like ocean water that has come in, and so they can't survive in those two locations. And so it's suggested to be habitat loss, but not just habitat loss from humans. Well, directly, right? So one was habitat loss. The other one is that there was a new species introduced. This is a different species entirely that was not native to Madagascar, was introduced. And it turns out, based on the really old paper from the 90s, that invasive species was already there in the 90s. And so that may have been what also drove it to extinction. So it's a combination of factors. Again, it may have been, definitely with the climate change and the loss of habitat, because as they're analyzing the location, like this habitat is fundamentally different from what has been described in the literature from when they originally looked. And then the second really important batch of information, right, is there was this invasive fish that was already present at high levels in the 90s. And that invasive fish is still around. So it's not that the fish themselves could not survive, right? It's that they were taken over at or the hypothesis they were outcompeted, number one, and then number two, some of their locations that were freshwater, like Sam mentions, if they're getting infected or like covered in salt water, then that's kind of, that habitat is lost to them. And then they said the deeper inlets have actually started to dry out. And so if you have less habitat and an invasive that does better, that invasive is going to outcompete the local fish or other fauna as well. And they did make an interesting comment in the paper of who knows what other fish or animals in this environment have been driven to extinction, which I thought was really interesting. That they don't know, right? But they're like, if there was a food chain, presumably there is, that depended on this very brightly colored fish, we could have lost those creatures as well, and we'll have never known about it. So, Chris, again, the wild thing to me in this study was when it started off, the whole premise was in the 90s, they found this brightly colored fish, and no one thought it was a new species. They were just like, oh, pretty fish, let me put it in a jar and move on. And now we're looking at it near 30 years later, and we're like, oh, shoot, this might be a fish that we have lost now. And it turns out we probably have. Looks a little... So, Cass, they did do a bunch of morphological analyses. They compared it to the existing fish in the area, like the close relatives. The biggest thing, looking at these holotypes, which are the representative species now what you measure against, they have like a different weight to those fins. They seem a little bit more elongated than those beta fish. On top of that, they've also got these, like, the lower fins in the males are apparently very, very unique when compared to other fish as well, like outside of this genus. This was one of the defining features. So these are small hooks at the base of the male fin. So this is not a reproductive organ. I know we've talked about a lot of differences in male reproductive organs and female reproductive organs, but this is that just a lower fin. And that lower fin, it has these little extra bit of hooks. And those extra hooks are an identifying factor that made them different. So again, it's not just one element of difference that was seen in the creature, but you're looking at multiple differences. Which again, I'm like, if all these differences are kind of adding together, it's really cool there's so many of them. right? And they're so structurally different. That's just not a fluke at that point. This structural differences are, true developmental differences, true differences in the genome of the creature, yielding to what is in fact a different species. So I was like, that is really, really cool. There's no video of them being alive. When we had them alive, no one knew that they were new fish. So there was just not documentation on like let's say if you hypothesized mating behavior, none of that, or defense behavior, or anything along those lines. None of that was actually studied here. I don't know, I picked this study because I thought it was really interesting in that it starts off with this story of like, here in the 90s we found this pretty fish. What does this actually end up meaning in terms of identifying a new species? And then we end up finding that there's in fact, no new species. Well, it is new, but we don't have it anymore and it's totally gone.